Yeah, that was very interesting and surprising initiative by biotech company that is developing an IRAK4 inhibitor. So IRAK4, it's becoming a really important target in leukemia, myelodysplastic syndrome, lymphoma, and potentially other diseases. To be honest, I only have knowledge on the MDS AML perspective of this. And the reason this is becoming interesting is for two reasons. Number one, this concept of what we call or refer to as alterations in innate immunity signaling that are common in these diseases. And IRAK4 is a central molecule on this pathway. So basically, by inhibiting IRAK4 downstream signaling, you may exert anti-leukemia or anti-lymphoma activity. So that's very exciting. And then more recently, it has been shown that perhaps specific subsets of patients with MDS and AML that have splicing mutations, they may have different isoforms of this IRAK4 molecule. And this could actually lead towards targeting of these agents, let's say IRAK4 inhibitors, for particular molecular subsets of patients that have a splicing mutation, because the splicing mutation is associated with a specific IRAK4 isoform that may be a more amenable target for IRAK4 inhibition, meaning that even if these IRAK4 inhibitors don't target the splicing mutation per se, maybe they have, as a consequence of the splicing mutation, this particular IRAK4 alteration that is more sensitive to IRAK4 inhibition. And this is very important because splicing mutations are extremely common in myelodysplastic syndrome and also in acute myelogenous leukemia. I'm also aware that these mutations are important in lymphoma, potentially related disorders like Waldenstrom's macroglobulinemia, etc. So this could have really significant interest. In addition, some of these IRAK4 inhibitors, by the nature of their design, may inhibit FLT3. So they may also be FLT3 inhibitors. So they may have pleiotropic effects. They may be able, IRAK4 inhibitors, I mean, we may be able to target them against a specific splicing mutated subset of patients, or maybe for FLT3 mutated patients. So this is an evolving field. And I think the seminar actually was very interesting, actually, because it covered a lot of these issues that I just tried to summarize in the last few seconds. So there are, as far as I know, there are 2 or 3 of these compounds that are either in the clinic or in development. So there's quite a bit of excitement about this. The more advanced of them is the one that you're referring, that we also know as CA4948, Emavusertib. And this data from the phase 1 trial was actually presented on a podium, as an oral presentation, I mean, at the EHA meeting in Vienna last June. And I think the data is quite exciting. So we have shown this at EHA also the past year, then there were updates during the year. But what we're seeing on the original, or not the original, on the initial phase 1 trial of this compound, is that we see significant anti-leukemia activity. So actually, a large majority of the patients had decrease in blast. This is unusual, actually, for phase 1 study, right, where we are looking for safety signal. And here, all of a sudden, we're seeing actually quite significant reduction in blast in patients that have HMA failure, multiple relapse refractory, maybe even treated with venetoclax. There were some patients with FLT3 mutated disease. So we think that for a phase 1 trial to see that level of activity is quite significant. And some of these patients actually went to achieve complete remission. So this is early in its development, but what we saw is very significant clinical activity for an asset that is very early on its clinical development. So we're excited, and now this is moving, hopefully pretty soon, into combinations of this IRAK4 inhibitor, perhaps with BCL2 inhibitors, maybe hypomethylating agents, and there are a number of other initiatives that are being discussed. I also think that these drugs could have, meaning IRAK4 inhibitors, could have a role in low-risk MDS. Indeed, actually, this pathway is also significantly engaged in patients with low-risk MDS. And there's a study in Europe by Dr. Platzbecker looking at the CA4948 also in this context. And there are other drugs from other sponsors that actually are also trying to target the subset of patients. So we may see this class of compounds both in low risk, high risk MDS, AML, maybe in combinations. Now, what we really need to do is actually find the proper dose and schedule. Because on the original design, we use, for instance, the CA4948 on a daily basis. I think that needs to be revisited. We need to think about 7, 14, 21-day schedule. So we need to figure out what is the proper dose and schedule, particularly as we move into combinations with these drugs. So the symposium, I thought it was extremely informative because it basically brought together like these three different areas of interest right now. So one was this kind of review of what innate immunity is, what IRAK4 protein does. That was extremely educative. The second leg of this is the splicing correlation, right? Like if you have splicing mutations like SF3B1 or some of the other genes, again, this may switch the conformation of these proteins, and that in itself was also really well described. And then the third was kind of like the therapeutic output of this, that is drugs that are being developed towards this pathway. And although this symposium was sponsored by a particular company called Curis that is developing this Emavusertib compound, the reality is that this was open to all the other drugs, and I believe they were mentioned, so it was not really conflicted to one particular agent, trying to give the the audience an idea of the compounds and the clinical context where these drugs are being developed. I thought it was extremely educative session, actually.